All right. So for today, I thought of talking about, my lab works on two different aspects which are linked by one particular thing, that is the endocrine pathway. Basically, we look at how chemicals interfere with male fertility. And the second aspect that we look at is how chemicals also affect our insulin signaling pathway that is leading to diabetes. So for today, I thought I'll just focus on one of the projects of that lab, which is reproductive toxicity. So before getting into reproductive toxicity, what I'm going to talk about will be in general, what is reproduction and why do we need to study reproductive toxicity, and how Drosophila would be a good model for reproductive toxicity assessment. This will be the structure of my talk, basically. Is that okay? I don't know how many of you are aware of Drosophila as a model.
Yes, sir.
Yes, sir. Yes, sir.
Yes, sir.
So I'll go, like one by one, if at all, if something appears to be very preliminary for you people, please excuse me, because I'm not very sure how many participants know about Drosophila and its reproduction or its fertility. So that is the reason I kept some of the basic things also before I get into my research component. Towards the end, like I'll be talking only about my research based on whatever that we have done in terms of the fundamental science. Is that okay?
Okay, sir.
Yes, sir.
Yeah.
Okay,
So
sir. Yes,
can you see my slide now?
yes, sir. Yes,
Yes.
sir.
Okay. So this particular one, as you can see here, this is the organization where I come from, that is the CSIR-Indian Institute of Toxicology Research, which is the only toxicology research institute are dedicated to toxicology in Asia, and it's second only to NIEHS, which is in US. So we look into several aspects of safety domain, basically how chemicals influence our health and what levels of exposure is safe and what will be risk or risky in terms of our health conditions, not only the human health, but at the same time, the health of the environment also. And apart from that, we also offer good number of services in terms of taking advantage of the expertise of the different scientists that are available to industry. And depending upon this is actually what we call it as a regulatory work based on whatever the requirements of the companies to get the regulatory approvals. There, whatever the different kinds of toxicity assays required, we provide those services to those industries depending upon what kind of products they want to bring it into my market, our market. And these are my contact details. If you want, you can note it down and you can contact me anytime later if you have any questions or queries. And my reason for showcasing these contact details is that I'm also in charge of skill development here. So if at all, if you want your students to be skilled in certain aspects or areas of biology or molecular biology or toxicology, you can always refer them to these contact details so that they can attend our programs. I'm not going to detail about whatever the programs, but you can get to our website to see what are the different skill development programs or the training programs that we offer, and the students can take advantage of those things. But for today, I'm going to just focus on how our lab has been working for almost like 2 decades now on understanding the basic processes of male fertility in Drosophila and how we translate that knowledge into reproductive toxicity or generating platforms for reproductive toxicity. So I don't know how many of you are actually teaching the basics of reproduction. So if anyone is there, can tell me like what is reproduction? No one? What is reproduction?
Is it sexual reproduction or asexual reproduction, sir?
Reproduction in general, ma'am.
Perpetuation of the offsprings to the next generation.
Yep,
by the male and female.
production of offspring with the
The
intention
production of young
of
ones.
yeah, production of young ones or production of offspring, and
It's called It is called the prodigation, I think.
See, the aim of reproduction is perpetuation, because everyone wants to pass on their gene pool to the next generation. So that is what is achieved through reproduction. So now ma'am asked whether are we talking about sexual reproduction or asexual reproduction. So for today's talk, it's going to be only on sexual reproduction, because Drosophila is a sexually reproducing organism. But before I get into the sexual reproduction of Drosophila, first I want to talk about the problem, that is the infertility. So if you talk about sexual reproduction, what is the ideal script of sexual reproduction? What do you consider to be the most ideal script as far as sexual reproduction is concerned? No one? What is the requirement for sexual reproduction?
Female gametes.
Gametes are required, sir.
Gametes are required, okay? And
Fusion of gametes, sir.
fusion of gametes, wonderful. But who does the production of gametes?
Organisms.
Organisms,
female organisms.
yes. So technically the ideal script is you have to have a male, you have to have a female, and they need to, for human concept, it's like they need to know each other before actually they think of getting into a relationship and producing progeny. Okay, this is the most ideal script in terms of successful reproduction, right? Especially like from biological point of view, getting to know each other, you have production of gametes and everything, but then until and unless there is fusion of gametes and passage of gene pool to the next generation, it is not considered to be successful. But not in all cases, you will see this kind of a successful transmission of gene pool to the next generation. But in some cases, you'll see that the script can go horribly wrong. I'm just showing you one example from, again, a movie. I don't know whether you have seen this one. Any guesses? Which movie is this? Any guess? Ma'am said like there are several people from North India. No, not Bajirao. Anyone?
Padmavat.
Padmavat. Yes. So you know what happens here? They get to know each other. That's fine. Like they get into a relationship. That's also fine. But there comes a disruptor, because of which at the end of the story, everything ends up in flames. There is no passage of gene pool to the next generation. So from the toxicology point of view, if you replace this particular disruptor with a chemical, what we call it as a xenobiotic, okay, which is a general environmental chemical, is referred to as a xenobiotic, because of which you can have a lot of problems in having offspring. So that is where the concept or the field of reproductive toxicity or toxicology is generated. The reproductive toxicity is this particular one, which is occurrence of any biologically adverse impact on the reproductive system or the functions of the reproductive system in response to these environmental chemicals. It can be male or it can be female. In male, it is referred to as male reproductive toxicity, which I'm going to focus today. And if it happens in female, it is referred to as female reproductive toxicity. And this can be anything in terms of effect on the reproductive organs per se, or effects on endocrine systems. And I'll just talk about endocrine influence in just a couple of minutes. And then the impact on the pregnancy outcomes. Any of these can fall into this area of reproductive toxicity, and the agent that causes reproductive toxicity is referred to as reproductive toxicant. That chemical or the agent which causes these kinds of changes in all the reproductive organs, it is referred to as toxicant, and generally you will see this kind of symbol on any of the bottles of the chemicals. Anyone like, I mean, you people are from masters or like, what What is the level in terms of colleges here?
Degree college.
Degree college. So do you have practicals also there, or like chemistry practicals or something?
Yes,
Or
sir.
any biochemistry practicals for that matter?
It depends on the semester and the syllabus.
No, that's fine. But then the thing
We
is,
have
if you people,
biochemistry
yeah,
also.
Yeah.
wonderful.
Yes, we have.
So if you go back to your labs and look for certain chemicals, okay, you will see this kind of label on those chemicals already printed, which means that particular chemical is potentially toxic to the reproductive organs. Okay,
By what way, sir?
sorry,
By what way it affects reproductive system, sir? Either inhaling or
hindering or
how it affects the reproductive system, sir?
I'll
Some
just-
chemicals
I'll
in
just
the lab.
I'll just come to it, man. I'll just come to it.
Okay, okay.
See, I just asked you as an example regarding laboratory exposure to chemicals, but whether you like it or you don't like it, current world is mixed with all these kinds of chemicals. Whether you like it or you don't like it, you are being exposed to different chemicals in your life now. Okay, whether you're using cosmetics or whether you're eating something or drinking something, you're always getting exposed to chemicals. And the toxicologist, the father of toxicology, Paracelsus, has defined it very clearly way back in 1700s itself, is everything is toxic. If you think, I mean, like, how many people think that water is safe? Everyone thinks water is safe?
Yes, sir, yes, but we
If
are
you-
drinking now RO water.
Yeah,
It
you
is
think
not
like
safe.
RO water is safe?
No, no.
No, sir, no.
No,
No,
not
there is
safe.
a
I'm not asking in terms of germs or anything. Assuming that your RO is very efficient in removing everything, do you think it is safe?
No, it is not safe.
Because even if it is like pretty healthy nowadays, you're getting alkaline purifiers and all that. With all that, if you drink too much of water, generally recommended is something around like 2 to 4 liters per day. And if you exceed that 2 to 4 liters per day, then it's going to put a lot of pressure on your kidney because it has to work overtime to remove whatever the excess water that is present in the body. So continuously drinking too much volume or quantity of water is going to cause toxicity. That's what it means, that everything what you are seeing right now or eating right now or being exposed to something is toxic, but it's the only dose which actually demarcates the safety from toxicity. As long as you are within the limits, you are safe. The moment you cross the limit, you are touching that limit of toxicology. So somebody asked me like, sir, how does it happen? The major mechanism that's been shown to be is the endocrine disruption, because majority of these chemicals, they mimic our endogenous hormones, estrogen, testosterone, all those. And they interact with those receptors and they trigger the activity of those receptors at times where they're not supposed to be present. And if you see, it's been like taught long back, even during BSc or even like intermediate nowadays or +2, it's being taught that reproduction is something which is very much regulated by your endocrine system. And there is always a positive and negative feedback loop that runs. So that way you have lot of opportunity for the chemicals to interfere with any of these processes associated with reproduction, leading to reduced fertility, be it male or female in this case. WHO has already recognized this as a problem. I mean, it's not just exposure to chemicals, but several other factors also kicking in, which is leading to increased incidence of infertility here. Just a second, I'll try to change the pointer. And as per the 2023 data of WHO, almost 1 in every 6 people is facing some kind of infertility issue at this point of time. And this is very clear when you go back to your own city and check how many infertility clinics were there earlier compared to how many you have right now. Almost in every nook and corner, you're going to get one infertility clinic these days, irrespective of the level of the whether it is a city or a town or a village, everywhere you have the branches coming up because of this major problem. And more or less, like as per WHO data, it is very clear that females and males contribute to, or problems from males and females contribute to infertility. And in some cases, it's like 1/3 cases, both male and female cases, or also male factors are also responsible for this infertility. But however, if you look at epidemiological data, that is the population-based data, if you see there is a severe problem that is affecting the males in terms of semen quality. Do you know what is the semen quality? Excuse me. Anyone?
In terms of motility, the
Motility.
structure,
Not just, yeah, in terms of motility, structure, and
Strength,
Sperm count, sir.
per counts, sperm count.
that's what it is. Semen quality again is defined by WHO in terms of 5-6 parameters. It is not just count alone. It is in terms of the concentration, how many million sperm are there per ml, and how many are having that normal structure, and how many sperm of those normal structure still have that motility. All these things define the semen quality along with the volume of semen. That is also is defined by WHO in terms of, for normal human beings, this is the standard yardstick that they have decided, that the volume has to be somewhere around like 3 to 5 ml, and the motility has to be at least 40% of them should have the motility, 50% should be having the normal structure. So it's like that. I mean, 40,000 million or 40 million per ml is the range which is given for normal sperm fertility, the concentration wise. So if you see the data here is in 1973, it was in the range of 101 million per ml. Whereas if you come to 2018, it's 49 million per ml, which is almost a reduction of 50% in terms of the concentration alone. I haven't put the data here in terms of sperm morphology because that's also being studied, and there is almost like 40 to 50% reduction in terms of the morphology also in these cases. And what is even more alarming in terms of sperm reduction is, if you see here, 1972 to 2000, the reduction was going at a rate of 1.16% per year, whereas 2000 onwards, it has become even more drastic that the sperm counts are reducing at a rate of 2.64% per year. So there is a big debate that's going on that if this particular trend continues, what will be the status of a male, whether there will be a male number one or male without maleness or the sperm production, how does it going to happen? It's a big debate that's going on. Let's not get into it. But what's important to see is this reduction in sperm counts has been associated with increased industrial production. If you correlate this way, the numbers are going down as the global chemical production or the load is going up. This is just with plastic production, but if you see good number of chemicals, there are several other classes of chemicals, this trend is pretty valid. So therefore, people have started looking at doing the toxicity, the reproductive toxicity assessment, which is pretty much using the animals. As you know, I mean, like all these animal-based studies are done with the expectation that you can extrapolate the data or the safety data to human beings. Because even though human is a very good test species, you cannot utilize human as an experimental organism for these kinds of studies because there are ethical issues associated with it. So you cannot use it for toxicity assessment here. So very much these animals are utilized for doing this reproductive toxicity. But the problem here is you have only 43% studies being extrapolated to human. Earlier it was studied that because of the similarity or conserved genetic constitution of mouse and human or rat and human, very easily all the results will be replicated in human. So the safety data would be very much relevant to human. But then the prediction wise, it's only 43% which turned out to be true in case of human beings. And the number of animals that are required for doing these kinds of toxicity tests, that's a big concern right now because almost 120,000 chemicals are there. And 80% of the chemicals do not have any data related to its safety or toxicity in terms of reproduction. And so if you want to assess those 120,000 chemicals, how many animals would be required for doing those ones? And in terms of reproductive toxicity, the time required to complete one study is almost 6 months per chemical. So 120,000 chemicals into 6 months, you can imagine how long this is going to take. And apart from that, there is always like these ethical issues that are concerned because human per se doesn't want to participate in these studies, number one. So when human himself or herself is not participating, what is the right of human to subject these animals? So that is where there is a major debate again, once again going on, like we should take away the use of all these animals from toxicity studies or experimental studies for that matter, and come up with these kinds of approaches where you can reduce the use of animals or replace the use of animals. The ultimate aim, actually it started way back in 1900s, even though this proposal came in 1959 to replace the animals, but because of the administrative problems and all that, it took a lot of time. But now they have started banning the use of chemicals. For example, in India, you cannot use animals for testing the toxicity of cosmetics. It's completely banned now. Cosmetic testing cannot utilize animals. It can only utilize alternate animal models or in vitro models or the cell lines, whatever that is. You can use it. And likewise, one after the other, for example, as teachers, you must be facing that problem. Earlier, there used to be these dissections and all that. Now you don't have those dissections in most of the colleges, as far as I know, because it's practically, it is like banned right now. In case of college or the experimental studies or demonstration purposes, you cannot utilize these animals for educational purposes anymore. So likewise, even in toxicology, slowly it's happening. So there is a lot of thrust in terms of generating new alternative methods wherein we can test the toxicity of chemicals, but at the same time, we can reduce the use of animals. So this is where we bring in this particular model system, that is a Drosophila, which we call it as half human, half insect. We call this as half human, half insect because at least 70% of the disease genes, human disease genes basically, are conserved in this particular organism. And being an insect, obviously its insect component has its homology to all whatever the beneficial or detrimental insects that we have in the environment. So in both ways, this model will be very useful in terms of studying toxicity using this particular one. So if you look at the Drosophila script of sexual reproduction, is this clear in terms of why we use Drosophila as a model now? The rationale for bringing. Hello?
Okay, sir. Sir, but Drosophila is also an living organism, no, sir?
No, ma'am. This is
Maybe
what
a
it
same
is.
type
This
of
is
reproduction, sexual reproduction.
Yeah, I'll just come to that, ma'am. It has sexual reproduction. See, the thing is, Drosophila, you say that it is an organism, but then the thing is, this you can produce in large numbers, number one. Unlike the animals, where, I mean, like if they are suffering, you will get to see that they are suffering. Whereas in these ones, it's very difficult to recognize that whether they're suffering or not. That's number one. We have to go through the ethical clearances and all that, because it has a nervous system, so pain sensing mechanism may be there. I mean, so far nothing has been reported with these organisms in terms of pain. So in that case, that is what is like all these lower organisms. Oh my goodness, somebody is moving it or like it's moving. Yeah. So that sensation, pain sensation is not recognizable. Therefore, the cruelty part of it is not pretty obvious, and mostly all these ethical considerations come into picture where there is a definite established vertebral system. Okay, when you have this, I mean, mainly for the chordates, this has been a big issue, but for invertebrates, they said, okay, fine, you can utilize these or develop these as alternate animals, because you can have large number of animals produced in your own lab and then you can use it. So as I said, coming to that script, it is you have a male and a female. They do get to know each other, what we call it as courting or mating or whatever it is, and which ultimately culminates in copulation. And subsequent to that copulation, females start producing a lot of offspring. And the numbers here go to any guesses how many numbers you can get from one particular mating or copulation? Any guess?
More than 12 to 15, sir.
12 to 15, because I put 12 to 15 here, so you're saying 12 to 15.
No, sir. No.
Maybe in hundreds.
It is in hundreds. In 10 days, generally, like I'm just telling you, like what we get in our lab. In 10 days, if you start with one pair, that is one male and one female, and allow them to copulate only once, that number is going to be around 300. Okay? So you can imagine if you have like 20 or 30 males and females in one bottle, how many progeny you're going to have. I mean, like the numbers is not a restriction here. And apart from that, this model has a wonderful collection of all kinds of molecular tools that you can imagine, with which you can manipulate the genome of this fly or generate transgenics in this particular model. So it's been the Cinderella of genetics, okay, for understanding the fundamental concepts of biology. So we continue that in our lab in terms of identifying the fundamental biology of male fertility in Drosophila, and then we translate that knowledge into screening platforms or platforms that are relevant to toxicology. So I'm going to give you initially in terms of, I'm going to talk about initially in terms of how we have identified different molecules that are essential for male fertility in Drosophila. And then I'll talk about how we translated this to reproductive toxicity in generating a Drosophila-based model for male reproductive toxicity. I'm going to talk about with the perspective of human health. So let me go with the first one, which is the male fertility. So if you dissect out the Drosophila male reproductive system, this is how it looks like. So like in any other like human being or like mammals, you have these coiled testes. In mammals, you don't have this coiling, but you have a testis. So here it's a coiled testis, and then this particular one produces sperm. And if you see here, this is labeled as seminal vesicle, and this seminal vesicle is the place where sperm is stored. And this this tubular one, the pink one, is what we call it as accessory gland, which produces majority of the seminal fluid proteins in Drosophila. So at the time of mating or coitus, the sperm are discharged from here into this ejaculatory duct, and at the same time, accessory glands also release their secretions into this one, where the semen is formed, and then through this duct, they pass through into the ejaculatory bulb, and here the pressure is created so that the semen is discharged to the female, which is used for fertilization purposes and all that. So if you see actually under the microscope, this is how the real picture looks like. You have this coil testis and the loops of accessory glands, the duct, and then the ejaculatory bulb. So what we asked is, okay, fine. How can we study the contributions of different molecules in this reproductive system? How do they contribute to production of sperm, or how do they contribute to production of seminal fluid proteins? So we took 37 genes which are associated with seminal fluid proteins and 18 nuclear receptors. Okay, I mean, like I said, estrogen receptor and androgen receptor. So equivalents of those receptors are present in Drosophila, and 18 of those are present. So we took all those 18. And next thing, what we looked at is, okay, what is the homology of these ones? And if you can see, you have, these are the ones, different nuclear receptors, which belong to the superfamily of steroid receptors, whether it is NR family, NR2 family, or NR3, 5, or 6. So these 18 fall into the same 6 families of vertebrate nuclear receptors. So what we did is we thought of knocking down the expression of these genes and then see if at all that will cause any defect in their fertility. So have you heard of this term RNA interference? RNA interference, siRNA, miRNA, those kinds of words?
Yes, sir. Yes.
Okay.
They shut down protein translation.
Not protein translation. Okay, we can say translation because it's actually post-transcriptional regulation, because of which translation doesn't go further.
So replication of DNA.
No, ma'am. It's not replication. It's after transcription. Once the mRNA is made, it has to undergo maturation, right? Before maturation, there are these ones which, I mean, basically the miRNA or siRNA, which will recognize the regulatory regions in the mRNA, and then whatever the double-stranded RNA that is synthesized or formed because of that binding of that miRNA or siRNA to the parent RNA, that will lead to destruction of the RNA. This is actually the viral mechanism.
Convey message, no, sir.
They don't convey message as such, but the moment they go and bind to the RNA, it is a trigger for the system to say earlier retroviruses were there, right?
Yes.
Earlier. So those retroviruses, they were RNA based, and the moment they find our DNA or like bind somewhere, it used to be a double-stranded RNA. That's what they used to make. So our endogenous system has a protecting mechanism that wherever they see this double-stranded RNA, the trigger is degrade that double-strand RNA. So that is how the knockdown mechanism works. So the miRNA or siRNA, which can be endogenous or through transgenic means, you can introduce that required ones. For example, I said 37 genes of SFPs and 18 nuclear receptors. So we design these miRNAs specific to those particular genes or the RNAs of those genes, and then we introduce them into the flies using this particular one, what we call it as GAL4 US system. Anyone like who has studied yeast transcription mechanism here? Yeast transcription or galloperons. Basically, this one is from the yeast system. GAL4 is a transcription factor which binds to upstream activation sequences, which is the promoter region, and drives the expression of whatever that is given downstream of this US. You put anything downstream of US, this particular, the moment you express GAL4, it's going to initiate the expression of that one. Take my word that way. And what we do is we put a promoter right next to GAL4. We make a fly from that one. This promoter will be specific to the reproductive system. It will express only in the reproductive system, which means you're going to generate GAL4 in the reproductive system, and you allow these to mate with the another female or a male, which is having US with a gene double strand RNA. Okay. So basically, when these two crossed, you have a progeny wherein these two come together. And in one case, you have the GAL4 with US gene double strand RNA. And in another case, you don't have the GAL4. You have the double strand RNA, but it doesn't get expressed because the driver is not there. GAL4 here is the driver which drives the expression of the US gene. Okay. So this we say is the one which is going to induce the knockdown. So there will not be any protein or intended protein here. Whereas this one will have normal levels of protein because there is no driver along with the double strand RNA. So we consider this as control, which has the normal level of protein, and this one as experimental. Is that clear? How do we generate control and experimental ones now? The one which is knocking down the gene expression, the one which is having the normal level of gene expression. Is that okay? Or any difficulty in understanding here?
Again, repeat, sir. Not clear.
Not clear. Okay. So I said GAL4 is something which binds to US. Right? In E system, they are traditionally trained that way, that GAL4 has to go and bind to this particular one, this particular promoter sequence, and initiate the transcription of whatever that is downstream of it. Okay. So
Okay.
what we do here is we express this GAL4 under the control of a promoter, which is found in the reproductive system. Okay. That way we have GAL4 only in the reproductive system. All right. And in the reproductive system, this GAL4 is going to go and bind to this US and will create or start producing the double strand RNA of whatever the one that is going to bind to your parental gene. Okay. And when you cross this female and male, you have these two different kinds of males. Because we are looking at the male fertility, we look at only the males. We are not concerned about the females here because the driver that we are going to use is going to be a little complicated in terms of male specific driver, like male specific expression, in the sense it's going to knock down that gene only in the male reproductive system, not in the female reproductive system. Okay. So that way you can get a male which has the GAL4 plus the dsRNA. See, this is simple Mendelian genetics here going on. This gamete will have promoter GAL4 and this gamete will have US gene dsRNA. So when these gametes fuse together, okay, you give rise to this particular progeny which has GAL4 plus dsRNA. Okay. Whereas here, if you see, there is another one here which is written as stubble, which is produced as a other gamete. And this gamete, when it meets or fusions with this particular gamete, you have this particular progeny coming in, which is US gene dsRNA with stubble, where there won't be any expression of this one because GAL4 is not there. Is that clear or still confusing?
Okay, sir. Okay.
Yes.
Okay. So we take these two and set up what we call it as fertility. We just first initially what we look for is, now we have reduced the expression of one particular gene in a male. Okay. So whether that reduction or reduced expression of that particular gene has any impact on its fertility. How do you test it? You take It's the same thing. Promoter gal 4, and it's coming up again. Wait, yeah. So you take the female. Okay, here you have this control or knockdown male. You take the female, put them into a vial. Okay, this is the food vial that we use for culturing or like setting up any of these meetings. So you put the female into this and you introduce either control male or knockdown male into this one. Okay, in the other vial, what you do is you introduce, if you're introducing control here, if this is the control, if you're in the other vial, you're going to introduce the knockdown male. And you allow them to mate, and once the mating is over, you're going to remove that male and you allow this female to start laying eggs for 10 days. And after 10 days, you remove the female and you allow those eggs to develop to adult stage. And then once you have the adults coming out, you'll start counting how many progeny have come from this particular mating. And you compare control to the experimental. The experimental one is the one that we have reduced the expression of the transcript levels of that particular gene. Is that clear? How do we do this fertility?
Yes, sir. Yes.
Okay. And by doing that, what we identified is that these many seminal fluid proteins are associated with male fertility. If you lose any of these seminal proteins in the fly, then these males will have either reduced fertility or it will lead to male infertility in these ones. And under the nuclear receptors, these are the two that we found. I'm going to talk a little more about these two because I'll be focusing on the endocrine disruption. One is ecdysone receptor, which is the insect-specific steroid receptor. And the other one is estrogen-related receptor. This is the most primitive estrogen receptor one can consider because from this only the origin of estrogen receptors happened later on in higher organisms. In Drosophila, there is no estrogen receptor, but it has only the estrogen-related receptor. So we further looked into its functions of like how do they affect male fertility. I can skip this one. This is just to show that whatever the seminal fluids that we identified has functions similar to the ones which are reported in mammalian models. Okay, so with
the knockdown experiment you conducted,
Yeah.
were silencing different particular factors, or all the factors are silenced altogether?
No. See, this is what the beauty of the Drosophila system. Here you can knock down or silence individual genes in specific tissues.
Okay.
For example, we silence the gene in testis alone. In accessory glands, those genes are perfectly normal.
So this means it would have been a repeated number of experiment or single experiment this
No, it
knockdown
will be a repeated number of experiments. It won't happen in the single one because each fly will carry only one particular gene silencing. Each fly.
Thank you, sir.
Okay, so you have to do those fertility assays I have shown you now. Oops, sorry. These fertility assays I have shown, right? So
Yes.
these ones you have to conduct individually. For each and every gene silencing, you have to have a control and a knockdown. The silenced one and the one which is expressed.
Yes, sir. Got the point.
Okay, so for like if I say 35 plus 18, which is 53, that means you have to do 53 fertility assays separately for these ones. And each one is going to have at least 20 to 30 replicates. It's not like individual, it's like 20 to 30 replicates you have to do to convince the reviewers that yes, indeed, this gene has something to do with that particular one. See, that is what the beauty of the Drosophila system, as you asked. That silencing can be tissue specific, or it can be timed also in the sense like at what stage of development you're going to induce the silencing. All these can be done through genetic means. You don't have to have any kinds of chemical to be added or anything. So that is how it's been very popular in terms of doing these kinds of genetic screens, in terms of be it like any parameter, be it fertility. I'm just showcasing the versatility of the model in terms of fertility analysis. Okay, so coming to ECR, the egg dyson receptor. So when we knock down the ECR, what we saw was there is increased apoptosis in the tissue. You know what is apoptosis? The programmed cell death.
Yes, sir.
Okay, so because of which you have, I showed you that accessory gland which was full of secretions earlier. Now the knockdown one looks pretty much like this, with some kinds of vacuoles, which you can see. This is the transmission electron micrograph image, where you will see these kinds of vesicles, which are pretty much empty. Nothing is there in these ones to say that there are proteins, and which we checked further to see if these proteins are being expressed. This is through Western blotting, where you check the expression of specific proteins using their antibodies. And if you can see in the controls, you'll see the expression here, whereas in the knockdown, you barely see any expression, which suggests that knockdown of ECR is leading to loss of seminal fluid proteins, which ultimately gets reflected in terms of making that male as completely infertile. There is no production of progeny itself here. So this is one example. And the other example that we studied is of estrogen related receptor. Okay. This knockdown of or silencing of this ERR in the accessory gland had no impact. I haven't shown the data here. But if you compare the testis of the control to the knockdown, do you see a difference here between control and the knockdown?
Yes, sir.
Okay. So whatever this robust coil has been reduced into this tiny little testis. And what you have here is seminal vesicle, which is normal, but the testis is highly deformed. And we looked at it and then we found that normal testicular morphogenesis. I'm not getting into the mechanisms through which ERR regulates it, because I thought it will be too complicated for you people. But take my word, if you remove or silence ERR in testis, it's going to affect the development of testis. And what we looked at is, okay, fine, development of testis is affected. What about the functionality of the testis? The function of the testis is to produce gametes, and the gametes are sperm. So when we took the cross sections of the sperm, as you can see here, this is the sperm from control. This is the transmission electron micrograph once again. This is the mitochondria, major mitochondrial derivative. That's what we call it as. And if you see here, this particular tiny one is considered as minor mitochondrial derivative. And then you have this actin filament arrangement like a wheel, which is what we call it as 9 + 2 + 2 arrangement. What you will see here, 9 + 2 + 2. So that is the arrangement. If you have a cross section of the sperm tail, this is what you get in Drosophila sperm. So the difference between Drosophila sperm and mammalian sperm is, in mammalian sperm, you people must have studied something like a neck piece or the nabenkan, right? That is where the mitochondria is. Whereas in Drosophila, throughout the length of the tail of the sperm, you have the mitochondria present, the giant mitochondria throughout that length. And you can see here as that giant one. And if you see here in the knockdown one, you'll see that this particular mitochondria size is reduced, and at the same time, you have the deformed 9 + 2 + 2 arrangement of that actin filaments. So everyone knows mitochondria is the powerhouse of the cell. So when the energy is less for the sperm, what do you expect? You expect sperm motility to be compromised, right? When there is no energy, like for example, if you haven't had if you haven't had your food, and if somebody wants you to run, how will you run? Okay, it will be very difficult to run because your energy levels are pretty low. So that is what we wanted to check. If that happens in the knockdown, what you're seeing is the live images. When I move the cursor, it will start playing, and wonderful. For some reason, this is not playing. Let me check. Yeah. Can you see the sperm moving now? You'll just see the green ones moving here and there.
Yes,
Just
sir.
like a highway. On the highway at night, if you see the cars going around, they'll be moving like this. This is what happens in the control conditions. If everything is fine, this is how they run. And if you see in the knockdown, do you see them moving? A very few are moving, and most of them are very slow or stagnant. Okay? So which suggested that yes, testicular function is also compromised in addition to the testicular development, because of which the sperm are having the reduced motility, which can lead to reduced fertility here in this case. Is that clear in terms of how we study or utilize Drosophila as a model for infertility in Drosophila, be it seminal fluid proteins or in terms of the sperm or testicular parameters? Is that fine?
Yes, sir.
Yes, sir.
Okay, because I'm going to switch now to reproductive toxicity of how we are exploiting these ones, because what I have shown you is there are conserved seminal fluid protein classes, conserved in the sense conserved between Drosophila and mammals, and there are also hormone receptors available which are essential for male-female fertility in Drosophila. Why I'm highlighting this one is when we actually started working with Drosophila as a model for reproduction or reproductive toxicity as such, there was a big question mark is, okay, in human, you have all these hormones, hormone receptors. Do you have anything in Drosophila of a similar kind? Do they perform similar functions? So it was very essential for us to show that yes, even in Drosophila, there are seminal fluids, and these proteins, like in mammals, they also regulate male fertility. And then there are hormones, and receptors respond to those hormones, and they also play a very important role in male fertility. So using that as the basis, we wanted to go with, okay, now you have this basic fundamental understanding that yes, things are similar to mammals. So why can't we translate that knowledge into a model for reproductive toxicity? And I'm just going to talk about one particular example of how we are exploiting this particular model as a tool for reproductive toxicity assessment with relevance to mammalian models. Okay. So in mammals, generally, whenever you talk about a reproductive toxicity, the traditional animal model based reproductive toxicity assessments include these different endpoints. One, they look for fertility. Sorry, somebody said something. Can I continue?
Yes, sir.
Yeah. So they look for changes in fertility of the male, or they study the sperm morphology, and they also count the number of sperms, and they look for the motility of the sperm, and at the same time, they look for the expression of reproductive genes and the genes associated with reproductive functions. And then they assess if the quality of the semen is compromised, and if it is compromised because of oxidative damage to the reproductive proteins or through endocrine disruption. So what we asked is, okay, fine, these things are the typical reproductive toxicity endpoints in mammals.
ಅದ್ರಾಗ ಎಂಟ್ರ್ ಆಗ್ಬೇಕು ನಾ ಎಷ್ಟಾಗೈತಿ ಇದು ಅದ್ರಾಗ ಎಂಟ್ರ್ ಆಗ್ಬೇಕು 5 ಗಂಟೆಗೆ.
Ma'am, Jayashree ma'am, please mute your system.
Sorry, ma'am.
Yes, sir. Yes, sir. Please continue.
So when we bring in Drosophila, one simple question that we asked, okay, fine, if these things happen in mammals, why can't we take one chemical which causes all these things in the male reproductive systems of mammals and see if we can find similar changes in Drosophila. So for that, we took this particular chemical, which is called as dibutyl phthalate. Okay. So have you seen these plastic pipes? How many of you use or utilize plastic pipes at home?
Yes, sir. We use.
Everyone, like more or less, like would be using the plastic pipes, right?
Yes, sir.
Yes, sir.
And some of the pipes are hard and some of the pipes are very flexible, isn't it?
Yes, sir.
So the flexibility there comes because of these phthalates. This is a class of chemicals. I'm just taking one example, dibutyl phthalate, but phthalate as such is one chemical class which are used as plasticizers to enhance the flexibility of the plastics. Okay. So, but then because they get in touch with all kinds of like our water, food matrices or whatever it is, they have the tendency to get leached into or released into those matrices through which we get exposed as human beings. And these are potential endocrine disruptors in mammals, because of which they are known to cause all these kinds of things. So what we asked is, okay, fine, we take this chemical and expose males to this particular chemical and see if it's going to cause these changes in Drosophila. So I'm going to go one by one in terms of how we do it. So basically the exposure wise, what we do is we first collect the eggs laid by the females, which are generally the fertilized ones that we take. We don't take the unfertilized eggs because we utilize females that are mated only. We don't utilize the virgin females. So we pick these eggs and put them into media that contain the chemical that we use, and depending upon what is the LC50 or the environmentally relevant concentrations, we mix that much with the food media and we allow these eggs to develop right from that egg stage to the adult stage. And then we take these adult males and then we start looking for all kinds of different parameters that I said. First thing I said is fertility. So when we looked at the fertility, what we saw was, this is the control. See, in all these experiments, it's very essential that you need to have a control which is not exposed to the chemical, which means the males which are derived from normal food, not the chemical mixed food. That you use as control and you take different concentrations. Toxicology doesn't run with one particular concentration. Until and unless you finalize that, yes, this is the effective concentration, you have to have multiple concentrations to show that what is the limit at which you have no effect and what is the limit at which there is an effect. Okay, depending upon that one, you are seeing one particular terminology here, what is called as LOAEL. Anyone has heard of this term? Anyone heard this term LOAEL, or there is one more term what is called as NOAEL?
No, sir.
No, sir.
Okay. This one stands for lowest observed adverse effect level. Lowest observed adverse effect level. Okay. So this lowest observed adverse effect level is the concentration at which you first start or having an effect in response to the chemical exposure. The first concentration, like for example, we started something around like 10 micromolar concentration of the chemical. You don't see any significant change compared to control. Whereas here, you'll start seeing a significant change when you compare it to control. Okay. So depending upon the concentrations that you have taken, you can define this, we defined as 10 micromolar as NOAEL. NO is nothing but no adverse effect level. There is no effect, therefore it is NOAEL is 10. LOAEL is like from 100 micromolar onwards, you start seeing reduced fertility with or when the males are exposed to this particular chemical, which is dibutyl phthalate. Some reason it's okay. Now it's moving. And the next thing that we asked, okay, reduced fertility happens when Drosophila males are exposed to this particular dibutyl phthalate. The next thing that we asked is what is happening to the sperm parameters in these exposed ones. So what we do is we take these control and exposed male, you mate them with oregano females, which are virgins basically, and then you remove the male after mating, and then you allow the female to lay eggs, but here you do it at different time points. I'm not going to get into like what are all the different time points at which you do it. And you freeze the female at that particular time point and you dissect out the female reproductive system and the male reproductive system. And then you see what is the status of sperm or the spermatogenesis stages in these ones. In male, you look for the sperm production and spermatogenesis stages, whereas in the female, you start looking for the storage of sperm in the reproductive system. So as I said, we have these flies where the sperm are labeled with GFP. Okay, I mean, you people must have learned today morning I was, accidentally I got into this link and I saw that somebody was teaching you the histone bundling and all that. So you heard of these histone proteins, right?
Yes, sir.
Histone nucleic acid interactions, right? So
sir.
there in sperm, what happens is the sperm nucleus has to be condensed to larger or greater extent possible. So for that, histones are not permitting that condensation, required condensation, I must say. So what they do is the organisms, they replace those histones with other proteins, what are called as protamines. Okay, these protamines permit highest condensation possible with the nucleic acids, because of which you have those pointed or nuclear shaped or whatever it is, like hook shaped sperm, all those kinds of things are possible. So in this case, Drosophila sperm are these kind of needle shaped ones, and people have labeled that protamine with GFP. That is the reason why you get these sperm heads labeled with GFP. So you expose those males which are having these sperm tagged with GFP and then look for production. And as you can see here, you'll see that there is reduced production of sperm at this LOAEL concentration of 100 micromolar. And if you further look into stages of spermatogenesis here, this is one step before this production of mature sperm, and you can see here it is getting affected. Whereas if you go to further down, it wasn't getting affected, which means the effect is at a later stage when the flies are exposed to dibutyl phthalate. And the next thing that we asked is, okay, production is fine or little reduced. What is happening to the motility of the sperm? If you can see here, let me see if it moves. Yeah. Can you see that sperm are moving here?
Yes, sir.
Males?
Yes,
You will
sir.
see it. You will see it that the sperm in the controls are moving pretty well, whereas when you go to the exposed ones, there is big difficulty here that hardly you'll see that any sperm are moving here and there. So the next thing that we asked is, okay, now sperm production is compromised to a certain extent, and at the same time, sperm motility is also compromised. So what happens to the sperm when they are transferred to the female? Why we study this female thing is, I don't know how many of you have heard about sperm capacitation. Heard something related to that sperm capacitation? Anyone?
No, sir.
Okay. So literally, like, it happens in mammals that the moment sperm are transferred to the female, they have to become hyperactive, highly energetic, so that they can get to the ovum. Okay, so they attain hypermotility there. So we wanted to see whatever the one that is non-motile, whether they'll become hypermotile when they go it into for the female. So controls, as you had seen earlier, you will see this movement pretty well because everything is normal here. They're not exposed to the chemical. Whereas if you see in the chemical exposed ones, you see some of the sperm, they're moving pretty well, right? Just like controls. But you see or look at these ones, these particular ones, this particular line, they're all clumped together and they're moving together pretty slowly. This clumping itself is shown to cause infertility because the sperm are no longer can be used for fertilization purposes. So it was very clear that exposure to chemical, like in mammals, affects the sperm counts and motility. We didn't see any changes in terms of morphology in our model in response to this dibutyl thalate expression. And then the next thing that we asked is, okay, fine, what is happening to the genes that are expressed in the reproductive system and whether there is reduced semen quality in terms of SFP production or their transfer. So we did, this is a simple PCR, semi-quantitative one, where we checked the expression of these different genes which are associated with production of seminal fluids or expressed in the testis and involved in testicular functions. So as you can see here, you'll see that the expression levels are going down, which is further clear when you see there is significant downregulation. This is through real-time PCR, wherein you can quantify the relative transcript levels relative to control when I say relative transcript levels. And you can see majority of these genes are significantly downregulated. So again, like Drosophila genetics come into picture, wherein you have the seminal fluid protein, which is labeled with GFP. And what you're seeing here is control with GFP labeled seminal fluid protein. And these are the ones which are exposed to chemical, where you'll see reduced intensity of the GFP. Here you can quantify that one. This is a semi-quantitative method, again using softwares that quantify the intensity, and you can compare and see whether it is significantly reduced. Or you have antibodies for those seminal fluid proteins, and you can check if the expression or the levels of the seminal fluid protein is going down, and then you can quantify if it is going significantly down or not. And the other thing that we did is, okay, fine, production is compromised, as you can see here. So how much of this is being transferred to the female when they are mated to females? So when we did that kind of comparison, as you can see here, control transfers this much, whereas as we increase the concentration of the chemical, there is reduced transfer of seminal fluid protein 2 in these cases, which suggested that not only the synthesis of seminal proteins is affected when the flies are exposed to DBP, but their transfer is also compromised when the DBP exposure is there. So it's pretty clear that genes or expression of the genes associated with reproductive functions are compromised, and semen quality is also altered in these cases. So the next thing that we asked, okay, I mean, like one of the mechanisms that people have shown in mammals is through oxidative modification of proteins or oxidative damage that these chemicals can induce in the reproductive organs. So can we detect such kind of modifications in our reproductive system? So in the Drosophila reproductive system, the method that we use to detect is oxyblot. So this oxyblot, what it does is it has this chemical that's called as dinitrophenyl hydrazine, and you treat the proteins with this dinitrophenyl hydrazine, and wherever there is oxidative modification, this DNPH gets converted into dinitrophenyl hydrazone, which is detected by this particular antibody, what is called as DNP. So when you look at this one, whatever the signals that you are seeing here, the black ones that you are seeing here on the Western blot, they refer to the proteins which are oxidatively modified. So through this way, we can recognize that in the controls also, there is subtle amount of oxidative modification of proteins, but with exposure, you have increased levels of oxidative modification of proteins in this exposed flies. So which suggested that yes, there is oxidative damage of proteins here also in Drosophila system when they are exposed to a dibutyl phthalate. The next thing that we asked is what happens to endocrine functions. So I told you, like when we did the functional analysis, we found ERR being associated with male fertility. And when we looked at the transcripts of ERR in controls and flies exposed to DBP, you'll see that there is significant reduction in terms of transcript levels. And at the protein level also, you can see that the moment you bring in chemical into picture, there is reduced level of protein, that's ERR, both in sperm sample and testicular samples, if you see it here. So the next thing that we asked is, can we translate this, or taking this particular candidate as an advantage, can we translate that into a screening system? So again, we took the advantage of Drosophila genetics here. I introduced you to one of the one like GAL4 UAS, right? Remember that GAL4 UAS initially I said?
Yes, sir.
Okay. And how many of you have heard about chimeric proteins? Chimeric protein. When you say chimeric protein, what do you understand? Chimeric protein is basically a combination of two different proteins. Okay. So whenever you talk about transcription factor, basic fundamental genetics, going back to that one, it has two different things. One is a DNA binding domain, and the second thing is transcriptional activation domain. DNA binding domain, BD, which is referred to as, and the transcriptional activation domain. And in case of steroid receptors, it is referred to as something called as ligand binding domain, LBD. Okay. Without that ligand binding domain, transcription cannot be initiated, because that is where the hormone will come and bind to it, and then it will trigger conformational changes in the DNA binding domain to initiate the transcription. Okay, so what people have done is they have taken GAL4, as I said, is also transcription factor. So they have taken the DNA binding domain of the GAL4 and they have included the ligand binding domain of ERR. Okay, which you can express under the control of heat shock. That means you give heat shock to these flies, then immediately they'll start producing this chimeric protein of GAL4 DNA binding domain and ERR ligand binding domain. Is this clear? Is this clear or very confusing?
Clear, sir.
Okay, so this one, when it goes and binds to this UAS, it's going to initiate the transcription of this reporter gene, what we call it as LAGZ. Some of you must have heard of this beta galactosidase assays or blue-white colony assays, right? You give or you do the X-gal staining and you see whether there is blue color wherever there is LAGZ or the beta galactosidase expression is there, right? So in this case, what happens is this is ERR is a constitutive receptor. So obviously, like in normal conditions, you always have the expression of laxi. And if there is any interference, if the chemical interferes with this process, then you will not have laxi, or you will have reduced blue color in this one. So when you go to that picture, whatever the blue color that you see here, can you see these blue spots? These kinds of blue spots here?
Yes, sir.
When you stain the tissue with X-gal, wherever that ERR activity is there, you will see these kinds of blue spots. In the control, you will see lot many blue spots like this, which means in the testis there is a lot of expression of ERR and the activity of ERR here. Whereas if you see the chemical exposed ones, you will see the number of the spots and also the intensity of the spots is going down, which is pretty obvious here when you see it here. Very obvious that the number of blue spots, just give me a second, sorry. Hello? लैब में कल दे देंगे, ठीक है, कल दे देंगे आपको. Thank you. See, this is another professional hazard. Like when you are sitting in front of the desk itself, then obviously you're going to get these kinds of phones. So these blue spots, reduction in these blue spots are reflection of interference of this DBP with the activity of ERR. And you can say that this particular one is a disruptor in the regular hormonal signaling of ERR. So in this case, I can say that DBP, like in mammals, causes endocrine disruption even in flies. Do you still have time, or like do you want me to go to summary? Because I think
you have to go to summary because it's time up.
Okay.
Raviram,
Fine.
because you have still some time to go questions.
Discuss. Okay.
Yeah.
Fine. I will then like skip this mechanism part. Okay. And yeah, so we could show that this particular model is much more sensitive compared to the higher organisms. That way, we projected it as an alternative, which has been now, at least the findings, not as a model, but the findings of the studies have been included in the regulatory documents to show that diabutyl phthalate causes reproductive toxicity irrespective of the taxonomical hierarchy, whether it is higher organisms or lower organisms. The reproductive toxicity of diabutyl phthalate is very common in all these cases. So I have told you that we have these two receptors which are very essential for male-female fertility, and I have shown you that at least with one particular receptor, I have shown you that we have a molecular tool with which we can detect the reproductive toxicity or the endocrine disruption potential of the chemicals. So although like I talked mostly about male fertility, the work is mostly done by the females, and ironically the work is done by two Snigdhas. One is Snigdha Mishra, who has done the initial part of identifying the endpoints and the fundamental aspects of molecules associated with fertility in Drosophila, and Snigdha Gupta is the one who has done majority of those DB